This program is a presentation of UCTV for educational and non-commercial use only.
Describe today is our efforts to understand how different parts of the cerebral cortex, are patterned. I first want to show the people in my lab who did all this work. But many of you probably know that the cerebral cortex, it segregates different functions into different parts. So back at the back of your head is where vision is processed. And then moving forward is where you hear. This S region is where you feel or touch sensations, metasensation. Moving forward, more forward is the motor cortex. And then at the very front is the nerve center of your nervous system, your prefrontal cortex, where you process complex information and make important decisions. And we're interested in the molecular mechanisms that generate this pattern of spatial organization of functional areas within the neocortex. More recently, we've focused on the prefrontal cortex because of its obvious importance for psychiatric illnesses. When we do these kind of studies, we really go very far back in time in embryology. And so this is half of a telencephalon. This is the cerebral cortex, and this is the basal ganglia. And what this color is showing is the molecular pattern expression of a transcription factor. And this transcription factor is expressed very high in the ventral cortex and very low or not at all in the dorsal cortex. And we believe that these patterns or gradients of molecular expression are fundamental to generation of spatial organization within the central nervous system. So what I'm going to tell you a little about today is what generates molecular spatial pattern in the developing cortex. And in this case, these are the progenitor cells and stem cells of the cerebral cortex. So what generates spatial pattern within the onloga or progenitors of the neocortex? And another question that I'll address is what generates the pattern of generation of neurons over space and time? So just as there's molecular pattern within the progenitor cells, there's also a pattern in the rate at which neurons are made. So this black marker labels the neurons, and you can see ventrally there's a lot more neurons than dorsally. So in the embryonic brain, or in the embryonic cortex, ventral regions make neurons much earlier and more efficiently than dorsal regions. And we think that this fundamental process is also important in building the differences between different regions within the brain. So to address these questions, I'm going to talk about primarily just four molecules that will be making manipulations of their expression. One is a transcription factor called COUP-TFI. This is a nuclear receptor transcription factor. It used to be called an orphan nuclear receptor because nuclear receptors are types of transcription factors that bind small molecules which activate their transcription, such as retinoic acid receptors, which bind retinoic acid. COUP-TFI is the same class of molecule as retinoic acid receptor. It was used to be called an orphan because it didn't have a ligand. More recently, it appears that COUP-TFI may in fact bind to retinoic acid. And then I'll talk about COUP-TFI's regulation, the expression of COUP-TFI gene by three soluble factors called fibroblast growth factors, or FGFs. And these three genes are FGF-8, FGF-15, and FGF-17. So these are the four actors in the story. So I introduced the idea of molecular gradients in the developing cortex, and it's recognized that some of the major transcription factors that pattern the cerebral cortex are all expressed in bidirectional gradients. So for instance, EMX2 is expressed high caudally and low rostrally, but is also expressed high dorsally and low ventrally. Whereas COUP-TFI is expressed as EMX2, high caudally and low rostrally, but opposite to EMX2, it's low dorsal and high ventral. And PAX6 is an additional variation on this theme. So we think that these gradients of expression are the fundamental ground plan for spatial information in the progenitor cells of the cortex that then generate the simple aspects of regional pattern and arealization of the cortex. So Andrea Faedo is the person who did the work on COUP-TFI function. Andrea's from Milano, and this is a picture of the central square in Milan, where he did not do the work. All right. So the conclusion that I'm going to tell you about COUP-TFI is that it does two things. It represses identity of rostral and dorsal fate. Said another way, it promotes a caudal and ventral identity of the cortex. And it also represses progenitor cell state. Said the other way, it promotes differentiation of neurons. The way we came to these conclusions is through changing the level of expression of COUP-TFI, either by overexpressing it using a transgene called the D6 COUP-TFI, or a loss of function analysis using a null mutation that was made by Ming and Sophie Tsai at Baylor College of Medicine. The overexpression assay was based upon using a enhancer region called D6, which was made by Stefan Kraus. And so we hooked the D6 enhancer promoter region 5' to the COUP-TFI open reading frame and made a transgenic mouse. And when we looked at the expression of COUP-TFI in the overexpressing mouse, we could see that indeed, compared to wild type, the D6 COUP-TFI mouse expressed a lot more COUP-TFI in rostral regions. So normally, the rostral region of the cortex expresses very low COUP-TFI, whereas in the D6 COUP-TFI you can see increased expression. Then Andrea looked to see whether or not the increased expression had an effect on the molecular patterning of the cerebral cortex. So here's the expression of COUP-TFI in the wild type and the overexpression. And to look at the molecular pattern, we used an analysis of the expression of PAX6 transcription factor, which is expressed high rostrally and low caudally. And when we overexpress COUP-TFI, in the region where there's increased COUP-TFI, there's downregulation of PAX6. And previously, we had shown that PAX6 is important for specification of ventral cortical identity. And so we think that this result is consistent with the idea that COUP-TFI can regulate cortical patterning through repression of PAX6. And when we look at other aspects of cortical patterning that take place later, when there are in fact neurons, not just progenitor cells, using a marker called cadherin-8, cadherin-8 is expressed in the superficial cortex, high in the rostral areas and low in caudal areas. When we look at the overexpression of COUP-TFI, which is still overexpressed at this age of development in this region, you can see that the cadherin-8 expression is reduced, making this middle region of the cortex have a molecular pattern more similar to the caudal region. So these overexpression studies suggest that COUP-TFI promotes caudal fate, and that's consistent with a publication from another laboratory from Michelle Studer and Dennis O'Leary, who used a loss-of-function mutation of COUP-TF1 and came to the same conclusion. Now I'd like to show you that COUP-TF1 promotes neurogenesis and represses proliferation. So here we are again back to looking at the ventral to dorsal gradient of COUP-TF1. This is in the wild type, and then this is in the overexpression mouse, where you can see that COUP-TF1 indeed is expressed much more in the dorsal cortex than in the wild type mouse. So then Andrea looked to see whether increasing COUP-TF1 changes the level of expression of an important regulator of the cell cycle, a gene called cyclin D2, which is expressed in a gradient as well, and most high in this middle region of the cortex. And indeed, in the region where COUP-TF1 is overexpressed, there's a very strong suppression of cyclin D2 expression. Consistent with that, when we look at the loss-of-function mutation for COUP-TF1, and we look in the ventral region where COUP-TF1 normally is most strongly expressed, you can see an upregulation of cyclin D2. Normally, it's very low in this region, so you can see it comes up nicely. Cyclin D2 is a nuclear protein which promotes mitosis or cell division. And consistent with this effect on cyclin D2 expression, we found an effect on the number of cells that were in the mitosis or M phase of the cell cycle using a molecular marker called phosphohistone 3 or PH3. So PH3 normally is found in the most immature cells in the ventricular zone, as well as in cells in the subventricular zone in the wild type. In the overexpression of COUP-TF1 mouse, Andrea found a marked suppression of the number of subventricular zone mitoses, and when he counted the number of cells that are undergoing mitosis in the ventricular zone, there's also a suppression of that number as well. On the other hand, in the loss-of-function mutation, you can see you can easily see an increased number of subventricular zone mitoses, and there's a subtle but measurable increase also in the ventricular zone. So COUP-TF1 represses cyclin D2 and represses the cell cycle. The other side of the cell cycle of a cycling cell is a cell that has left the cell cycle, in this case, at this stage in the development of the brain, a neuron. So we asked to see whether or not there were more neurons being made in this overexpression mouse. And for that, we used a marker of early neurons called TBR1, which labels the early born neurons in the cortex. And when you overexpress COUP-TF1, you can see about a twofold increase in the number of neurons that are produced, consistent with the idea that decreased cell division, and then the cells choose, instead of dividing or dying, because we don't see increased cell death, they choose to become neurons and differentiate. So in summary, COUP-TF1 is expressed high ventrally and low dorsally, as well as high caudally and low rostrally. But this ventral to dorsal gradient is nicely correlated with the normal maturational gradient within the cortex. We make more neurons ventrally early than dorsally. And I showed you pretty convincing evidence that if you increase COUP-TF1, you make more neurons. So how does COUP-TF1 regulate patterning of the cortex, this suppression of rostral and dorsal fate? And how does it control the switch between proliferation and differentiation? We have a little bit of information about this. And the way we got this information is we assayed some of the known major signaling pathways that are taking place in the embryonic brain. And these include the RAS and AKT signaling pathway, the Wnt signaling pathway, the sonic hedgehog pathway, and the TGF-beta pathway. We didn't see an obvious effect on either the sonic hedgehog and TGF-beta pathways, but we saw repression of both the RAS-AKT pathway and the Wnt pathway. Now, to show you a little bit of evidence for these conclusions. So what is the RAS and AKT pathway? These are signaling pathways that are downstream of receptor tyrosine kinases, as well as some other receptors, but primarily best known for receptor tyrosine kinases. And One of the most abundant of these families in the developing brain are the FGF receptors. So the receptor tyrosine kinases are transmembrane proteins that on their extracellular side bind the ligand FGF-8 or other FGFs, and on their cytoplasmic side can signal down the RAS MAP kinase pathway or down the AKT pathway, as well as down this PKC delta pathway. We can assay whether these pathways get activated using histological measures by using antibodies to phosphorylated ERK kinase or phosphorylated AKT substrates. And I'll just show you what happens when you measure phosphorylated ERK. So phosphorylated ERK is shown here in these little red dots that line the ventricle. This is in the wild type. In the overexpression of COUP, you can see that the number of cells that are positive for the phosphorylated ERK is greatly reduced. And in the loss of function of COUP-TF1, there's a subtle increase in the number of phosphorylated ERK positive cells. Andrea found a similar phenotype by looking at phosphorylated AKT substrate as well, but I won't show you that. So here's the receptor tyrosine kinase, and as I told you, it can activate the MAP kinase pathway in ERK. We can also activate the AKT pathway. The AKT kinase is also well known to integrate signaling this pathway with other pathways. For instance, it represses the activity of GSK3 beta, and GSK3 beta is an inhibitor of the Wnt pathway. So therefore, activation of AKT activates the Wnt pathway. So we assayed the Wnt pathway using a transgene measure of the Wnt pathway that expresses beta-galactosidase under the control of a TCF enhancer. It's called the BAT-GAL reporter. So in the wild type, you can see lots of blue cells. These are cells that are responding to the Wnt signal, whereas in the overexpression of COUP, there's a suppression of number of cells that are responding to Wnt. So currently we have a very simple model that COUP represses signaling in the FGF pathway, RAS and AKT, and the suppression of this might be the reason that there's less Wnt signaling, although it's very likely there are other more complicated and maybe more correct epistatic or arrangements of how these signaling pathways relate to each other. In any case, overexpression of COUP represses all three of these readouts, RAS, AKT, and Wnt. Now, the RAS pathway has been positively linked to rostral cortex specification and proliferation, and the Wnt pathway is linked to dorsal specification and proliferation, as well as caudal specification. So we've shown that COUP-TF1 represses this. It also represses rostral identity. It also represses proliferation. So this is a nice correlation that might explain why we see, with overexpression of COUP, decrease of rostral identity, decreased proliferation, decrease of dorsal specification as well. So COUP-TF1, we think, is an important transcription factor in patterning the cortex through repression of proliferation, promoting differentiation, and repression of rostral dorsal fate. So then it begs the question, what is it that regulates COUP expression to create this gradient of high COUP ventrally and caudally? So the next part of my talk will address what are some regulators of COUP. And we think one of the major forms of regulation of pattern in the developing cortex is through something we call patterning centers. These are part of the brain. They're embedded right within the neuroepithelium. There's one right up the front, which we call the commissural plate, that expresses FGFs. There's one along the dorsal midline and extending laterally into the chorioplexus area that expresses high levels of BMPs and Wnt's. And then there's a ventral region that expresses sonic hedgehog. So there's three different regions expressing these signals that can control regional fate, cell identity, and the switch between proliferation and differentiation. I'm going to concentrate on the function of the rostral patterning center and the role of the FGFs. And I mentioned 3 FGFs, FGF-8, 15, and 17. And the work on FGF-8 on rostral neocortical patterning was started in the mid-90s by Kenji Shimamura, and then extended in the early 2000s by Sonia Gurel and Elaine Storm, and then Ugo Borello finished up the work that I'll be telling you about. So FGF-8 expression begins very, very early. It begins during gastrulation, and it can be found in many parts of the gastrulating embryo, including the rostral end of the neural plate, which in section you can see here, this little tip region, which is the transition between the front of your brain and your skin. So FGF-8 is in a structure called the anterior neural ridge. We were able to study FGF function in vivo in the mouse thanks to mutations that were made at UCSF by Gail Martin's laboratory by Eric Myers and Mark Lewandowski. And they made an allelic series of the FGF-8 gene in which they introduced changes into the gene that sequentially decrease the amount of expression from it. And just want to introduce the names of some of these alleles. Here's the wild type allele. This is called the neo-allele that expresses about 30% the normal amount of FGF-8. Then there's a floxed allele that has 2 LOX P sites which enable us to delete exons 2 and 3 using Cre recombinase to generate the FGF-8 delta 2,3 or deletion allele. So here's a wild type telencephalon. And here's a telencephalon that has about 15% normal FGF-8 because it has a deletion allele and the neo allele. And you can see that it is very abnormal morphologically. It has holoprosencephaly. And we've shown that FGF-8 has a important interaction with sonic hedgehog. Sonic hedgehog, in fact, is required to maintain FGF-8 expression, and FGF-8 is important for expression of sonic hedgehog. And so either way, by reducing FGF-8 or sonic hedgehog, you can have a holoprosencephalic brain. Here is a mouse that has essentially no FGF-8, and the telencephalon, which is just this part here, is much smaller, and its dorsal midline looks like only choroid plexus. It's also holoprosencephalic. So these are subject of other studies that are in more detail about mechanisms of growth and patterning of the early embryo, but they point out that FGF-8 is extremely important in creating a normal forebrain and cortex. But it doesn't help us study cortical patterning because these animals have a very tiny cortex that won't let us ask what happens to the prefrontal cortex, what happens to the somatosensory cortex, et cetera. So for that, we utilize only the mice that have the neo-allele, which is a hypomorphic allele. It still expresses FGF-8, but just at a lower level. And so in these studies, Sonia Gurel compared wild-type mice to mice that are homozygote for the hypomorphic allele. And you can see that the size of the brain is only slightly reduced in this situation. So running on about 1/3 FGF-8 is enough to make a reasonable sized telencephalon. But if you look at COUP-TF1 expression, you can see that ordinarily there's very little COUP-TF1 in the rostral cortex, but now in this FGF-8 hypomorphic mutant, COUP-TF1 goes all the way to the front of the brain. And from what I was telling you about COUP-TF1 function earlier, where overexpression of COUP-TF1 promoted caudal fate and suppressed rostral fate, you could guess what would happen to this poor mouse's frontal cortex. So here's a molecular assay on the animal that shows that its frontal cortex indeed is really small. So this is just a stain for different regions of the embryonic cortex using ID2 as a marker. And the rostral regions have high ID2 in the superficial layers of the cortex, whereas the parietal or middle part of the cortex has very low ID2 in the superficial layer. And then if you look in this FGF-8 hypomorph, you can see that the size of the cortex is pretty normal. But this region that has low FGF-8 in the superficial layer has moved all the way to the front, just leaving a very small part with a frontal cortex-like molecular pattern. These animals died the day of birth, so we can't use more definitive markers of regionalization of the cortex. But they led us to hypothesize that FGF-8 or FGFs in general are important in allocating progenitor cells within the cortex to give rise to the proper balance of cortical areas. So ordinarily you have a frontal cortex of this size and a somatosensory cortex of this size, but if you have not enough FGF-8, you have a small frontal cortex and you have a rostrally expanded somatosensory cortex. And of course, this would not be a good situation if you wanted to have an animal that would be reasoning and not just feeling. So FGF-8 represses COUP-TFI, and COUP-TFI represses rostral identity. Let's try to remember some of these relatively simple relationships, because things get more complicated, and there are more than one FGF in the rostral patterning center. There's FGF-15, 17, and 18, in addition to FGF-8. And to make things a little bit more complicated, in humans, FGF-15 is called FGF-19, and I can't explain what happened with that nomenclature glitch. So this is the family tree of FGFs. There are 22 FGFs in mammals. There's the FGF-8 family, which includes FGF-8 and FGF-17 and FGF-18. That means these proteins are very similar. And then there's the FGF-19 family, or FGF-15, which is separate. And so the molecular organization of these proteins are, although they're similar enough that they're called FGFs, but they are quite divergent. And I should point out that unlike FGF-8, 17, and 18, which don't diffuse very far away from where they're produced because they bind very tightly to heparin sulfite proteoglycans, they kind of get stuck out in the extracellular matrix, FGF-15 is very soluble and can diffuse long distances, according to work of other labs. Okay, so here's the expression of the four FGF genes that I referred to. The FGF-8 family, 8, 18, and 17, are all expressed in approximately the same region. In fact, 8 and 18 look about the same in their regional pattern. FGF-17 is a broader expression pattern expanding dorsally from this region. So there's a core region where all three are expressed, but FGF-17 is unique in being expressed more dorsally. And then on the other hand, this weirdo, FGF-15, has overlap in the ventral region, but it expands much more ventrally. So let's find out what these different FGF genes do. I'll tell you about mice that have mutations in FGF-17 and FGF-15. So the FGF-15 analysis was done by Ugo Borello, who's from southern Italy. This is Naples. And the mice were made available to us by Kiesmer and John McWhirter. Okay, so look at FGF-8 and FGF-15 expression at a younger age. Embryonic day 9.5. FGF-8 is right here at the front of the developing telencephalon. That would have been the anterior neural ridge one day earlier during gastrulation. And then FGF-15 is expressed in an overlapping pattern in the front, but then expands much more extensively within the developing telencephalon. So many more cells are exposed to FGF-15 expression than FGF-8 expression. Now, if you remember, FGF-8 mutants had increased amount of COUP. FGF-8 is a potent repressor of COUP. And we were expecting the same result with the FGF-15 mutants, but we found the exact opposite. So at this age, COUP-TFI expression is found in the caudal telencephalon, which is this region here, and not the rostral telencephalon, consistent with what I've been telling you that COUP-TFI is not expressed rostrally. If you remove FGF-15, COUP-TFI expression goes way down. So FGF-15 is required to promote COUP-TFI expression. And when we look 3 days later in the developing cortex, here we see COUP-TFI high ventral, low dorsal, as I've said many times. But in the FGF-15 mutant, the COUP-TFI gradient becomes much more shallow. Still have it down here ventrally, but the level of expression dorsally goes way down. So indeed, FGF-15 promotes COUP-TFI expression. You may also note for specialists that the shape of the cortex is quite different. It looks maybe elongated in the mutant. So we have decreased COUP, and if you remember what happens in the COUP loss-of-function mutation, where we found increased mitosis, one would predict that you would see increased mitosis in the FGF-15 mutant if the COUP-TF1 expression is the driving force for the mitosis. And indeed, Ugo found a significant increase in the number of phosphohistone 3 labeled cells in the ventricular zone and subventricular zone in the FGF-15 mutant. So perhaps the change in COUP is one of the most important features here. And if you remember, COUP-TF1 gain of function caused more neurons, so loss of function would make less. And indeed, if you measure the thickness of the cortical plate, as Ugo did here, you can see a subtle but clear decrease in the thickness of the cortical plate, which means a decrease in the production of neurons. So our friend COUP-TF1, we know, inhibits proliferation, promotes differentiation, and represses rostral dorsal fate. And at least we've defined two important regulators of its expression, FGF-15, which promotes its expression, and FGF-8, which puts a brake on its expression. So we're beginning to understand how this COUP-TF1 expression pattern or gradient is generated, and then we understand a bit about how COUP-TF1 controls these important features of building a cortex. So FGF-8 and FGF-15 and COUP have very extensive and long-range effects on cortical patterning and growth. So almost the whole cortex is changed, which is very interesting and very important. But we know that there are many subdivisions within the cortex. How do you begin to build up smaller pattern within the cerebral cortex? Prefrontal cortex has many regions. In particular, you have the dorsal, dorsolateral medial, and orbital frontal. Is there a way to specifically build each of those three? And I'll show you some evidence that we think that FGF-17 is a way that that is done. So in the frontal cortex, as I mentioned, we have orbital frontal, dorsolateral, and then on the medial side, dorsal medial and medial. We were again lucky because we didn't have to make these mutants. David Ornitz's lab made FGF-17 mutants. And unlike the FGF-8 and FGF-15 mutants, which die on the day of birth, FGF-15 null mutants are viable and fertile. They have no apparent behavioral phenotype when you're looking at them, and their brain looks very normal when you look at them superficially. But fortunately, I had an outstanding graduate student, Jeremy Cholfin, and his wife, who didn't do any work, but she's a very fine obstetrician, who did the analysis of the FGF-17 mutant. So here we see the rostral patterning center, which expresses FGF-8 and FGF-17. And as I mentioned, FGF-17 expression is broader than FGF-8, going into more dorsal parts of the cortex. In the FGF-17 mutants, FGF-8 expression is still expressed, so we don't have to worry about the global effects of FGF-8 on growth and patterning of the cortex, but of course FGF-17 is gone. To find a phenotype, we had to be a little tricky, because if you look at the size of the cortex, it looks very normal. There's about a 5% reduction in surface area, we think, but sometimes it looks totally normal. So we built into this mutant a transgene that glows green because it expresses GFP under the control of the FA2 gene. So this FGF-17 mutant has this green marker of the frontal cortex. So this is a wild-type frontal cortex that expresses FA2, and this is FGF-17 frontal cortex that has less surface area that's strongly labeled by the GFP fluorescence. And then we found some other markers that would work for us to assess frontal cortex size. One is the transcription factor LMO4, which nicely labels the frontal cortex strongly here. And you can see that LMO4 expression surface area is also reduced. And when Jeremy did some quantitation of this, he found that the size of the frontal cortex using these two measures is reduced by about a factor of 2. And he sectioned the brain to see whether or not different parts of the frontal cortex are affected. And as I mentioned, the mouse frontal cortex, just like your frontal cortex, has a dorsal medial, an orbital frontal, and this gustatory or dorsolateral frontal cortex. And we had a hard time finding molecular markers for these. In fact, we don't have a definitive set. We have we're on our way to having some information about this. So for instance, neurogenin 2 is strongly expressed in the ventral medial and the orbital frontal cortex. And the FGF-17 mutant, we don't see any effect on neurogenin 2 expression in the ventral medial or orbital frontal cortex. And these are parts of the frontal cortex that you can think of as sort of limbic frontal cortex. On the other hand, the dorsal frontal cortex is very positive for LMO4, and that's greatly reduced. And dopamine receptor 4 is expressed in all parts of the frontal cortex, particularly orbital frontal, medial, and dorsal. And you can see that the part that's reduced is this dorsal and dorsal medial prefrontal cortex. So we think that FGF-17 has a relatively specific effect in reducing the amount of dorsal and dorsal medial prefrontal cortex that's produced, and doesn't seem to have an effect on the limbic frontal cortex, such as the orbital frontal and ventral medial. That's shown here in this schema, where the dorsomedial and dorsal, the red regions, become smaller. And what gets bigger, I'm not going to show you data for this, but it's very clear, is that the parietal cortex or somatosensory cortex expands rostrally into this region. So FGF-8 has long-range and local effects on transcription factor expression. I'm going to show you that FGF-17 primarily has local effects on transcription factor expression in the progenitor or stem cells of the developing frontal cortex. So here's our friend COUP-TFI, high ventral, low dorsal in a wild type. In the FGF-8 hypomorph, you can recall that COUP-TFI expression gets very strong, and you can see its expansion very robustly into the dorsal cortex in the FGF-8 hypomorph. Whereas in the FGF-17 null mutant, now this is a hypomorph and this is a null, has a subtle but increase of COUP-TFI. It does not allow COUP-TFI to come into the dorsal cortex as much as FGF-8. So FGF-17 has a break on COUP-TFI, but not a real strong break on it. On the other hand, FGF-17 seems to be more important for promoting the expression of other transcription factors. And one that we found that has this property is a transcription factor called ERM, which is high in the dorsal and medial prefrontal cortex. In the FGF-8 mutant, ERM expression is reduced in the dorsal, but not so much in the medial, whereas in the FGF-17 mutant, ERM expression is reduced much more, in my opinion, than in the FGF-8 mutant, both in the dorsal and in the medial. Unfortunately, we don't know what ERM loss-of-function mutants look like. Well, we do know, we've looked at the mutants, but we haven't found a phenotype, so I don't know what ERM does. But at least we can use ERM as a molecular assay to show that FGF-17 has a prominent role in controlling the molecular identity of this analog of the dorsal and medial prefrontal cortex. So COUP-TF1 promotes differentiation, suppresses proliferation, and FGF-15 and promotes the COUP-TF1 expression, and FGF-8 and FGF-17 suppress it. And we think that this property contributes to the gradients of neurogenesis in the developing brain, in which the ventral parts differentiate before the dorsal, and likewise the rostral differentiates before the caudal. I didn't show you that data. And we also think that this system regulates regional fate through suppression of rostral dorsal fate through FGF-8 and FGF-17 functions, repression of COUP-TF1. So in conclusion, FGFs regulate the relative sizes and natures of cortical areas. They do that through controlling the expression of COUP-TF1, but other transcription factors that I didn't talk about to keep this talk simpler, such as EMX2, PAX6, and a whole slew of other ones. And of course, this major effect on cortical patterning can be used during evolution to create different sizes of cortical areas as they turn out to be needed or useful to fill in ecological niches. But certainly if you don't have enough FGF-8 or COUP or FGF-15, you're going to have measurable effects on the nature and size of your cortical areas, and one can only speculate what the consequences these would be in terms of behavioral manifestations in humans. Although because the FGF-17 mice live, we've been able to study the effect of this through collaborating with behavioral laboratory of Leonard Mucke, and the work was done by Kimberly Scarce-Levy and Eric Roberson. And they threw the behavior book at these animals, and these animals were able to read the book, and they passed all the tests. But finally, Kimberly found something they didn't do very well. First, I just want to point out that in the adult brain, although they're looking very normal and the size of the adult brain is really normal, molecularly they're not normal. We can again use this FA2-GFP transgene to label the frontal cortex, and you can see that the rostral cortical regions has very little. So they don't recover their frontal cortex as they get older. These are 180-day-old animals. So here's a behavior assay in which the animals failed. So I call this the socially awkward mouse, because what happens in a normal mouse is if you put a girl mouse in a cage and then you put a boy mouse in the cage, the boy mouse gets very excited. Take the boy mouse out, and then you wait, some whatever time, 10 minutes, put him back in, boy mouse gets excited but less excited. And you keep repeating this, and you find that with repeated exposures, the boy mouse gets bored. That's not supposed to be funny. And then you take that girl mouse out, and you put a different girl mouse in the cage, and then you put the boy mouse in, and he's just as excited with the new girl mouse as he was with the first girl mouse. Again, that's not funny. No. The FGF-17 mouse, oh dear, I lost my light. There it is. This little pink dot barely shows a difference with the new mouse. He's able to recognize the old mouse and extinguishes his interest, but when the new mouse is brought in, he's not as interested in her as the wild type or heterozygote controls. So we think that this is evidence that the abnormality in the prefrontal cortex found in these animals is impeding the ability of the male mouse, to distinguish individuals. Likewise, that's the only assay I'm going to show. There's a couple other assays that also show behavioral phenotypes as well. So again, the part of the frontal cortex that's small in these animals is the dorsal medial, which is also called the anterior cingulate, which is very famous in neuropsychiatry. And some people can describe the anterior cingulate as being important in deciding the utility of behaviors, how useful a behavior is for the animal. For instance, how important it is to distinguish one individual from another individual, including social evaluation interaction. That's been studied in humans and primates and rodents as well, various ways, and it's been shown that anterior cingulate is very important in that. So we think it's probable that the reason our FGF-17 mutants have this problem in recognizing one mouse from another is that their anterior cingulate is small. So I'll end here. These are the people who, I think I told you everyone who did the work as we went along, and these are some of the other people who have been involved with the projects, as well as important collaborators and so on. And I'll open the lecture to questions now. Thank you.
Thank you, John, for that wonderful talk. And we'll open the floor now if anybody has any questions.
I don't know a lot about mice, but can you tell me whether that reflects an olfactory or a visual phenomenon? Mm-hmm. Your very last finding that you were talking about here.
That's a very important question. Mice are, as you're alluding to, very olfactory driven, and they're also very somatosensory driven. Those are the two senses that are the most salient for them. Then I would say probably audition and visual come in after that. We were very worried that this phenotype could be secondary to olfaction, particularly to the vomeronasal organ or accessory olfactory bulb. And so we did a ton of controls assaying their ability to smell various odorants, those that activate the main olfactory bulb as well as those that activate the accessory olfactory bulb, such as pheromones found in urine. And we found no evidence based on those assays that they have a deficit in any of those areas. Vision, we did not test. I'm not that familiar about the role of vision in how important that is in social assays for the mouse, however.
There must be changes in...
Let's use the microphone.
There must be changes in neural circuit and neural connectivity associated with these knockouts.
Yeah.
Can you talk a little bit about that?
So that's a very, very good question. Probably most of you who work on neuropsychiatry or neurology are interested in the role of an important circuit that connects the prefrontal cortex through the basal ganglia to the GABAergic parts of substantia nigra, to the thalamus, back to the prefrontal cortex. We think that circuit is essential for learning just about everything. That's my opinion. And learning complex patterns of things and putting them together in a way that's useful for the animal. So we looked specifically at the projections from the prefrontal cortex to the striatum using these transgenes that label the prefrontal cortex, the F8-2 GFP. And that because it's GFP, it can diffuse into the axons, so we were able to follow the course of the axons. And we got a little bit of information from that. We found that, you wouldn't be surprised, that there's decreased numbers of those axons because there's decreased expression of that gene. So it's a bit of a circular argument that there is a problem in connectivity. When we put dye into the prefrontal cortex to look at this, irrespective of the molecular nature of the cells, we found some evidence that the prefrontal cortex indeed is smaller through its reciprocal connectivity with the dorsomedial thalamus. But other than that, we didn't do a good job at answering your question. Karen.
So this regional and areal specification that you're talking about is happening early, and I'm just wondering, is there still any idea whether there's an influence of activity or innervation on that?
Yeah. So you're referring to the old nature versus nurture argument about how you make cortical areas. And there's no question that nature is completely essential in building the foundation of our cortical areas. That's without now a doubt. Nurture or activity is extremely important in making those areas work properly. If they don't get input, then they're not going to mature properly. The circuits don't mature properly, and you'll have a non-functional or poorly functioning visual cortex or somatosensory cortex. So late aspects of circuitry, connectivity, are completely dependent on activity.
John, did you look at the somatosensory cortex in these animals? In the one graph you showed that the parietal was expanded. What about the barrel field?
Yeah, so if you label the barrel fields with serotonin or cytochrome oxidase, you find that they move rostrally. We didn't do a good job determining whether or not the surface area dedicated to barrel cortex was changed.
So, John, these 6 mice that overexpress COUP-TFI at later stages, they have considerably smaller forebrains.
Yes.
Is this exclusively due to proliferation? It seems like that it affects also subcortical structures. Is this true?
Well, the D6 is only expressed in the cortex. And so the size of the brain is smaller. Is the size of the basal ganglia smaller? I don't know. We didn't measure that.
And do they live to adulthood?
Yes, they live to adulthood. They're walking around. They have this really shrunken down little cortex. And Andrea has evidence that that shrunken down little cortex has taken on more caudal fate. In fact, it looks like a poorly made hippocampus based upon a connectivity analysis he did where he put dye in and found that get reciprocal connections to the entorhinal cortex. We haven't published that, though.
Hi, John.
Hi.
I was wondering if you were able to dissociate the novelty component.
Yes. Yes, Kimberly and Eric did assay novelty to see whether or not the new mouse versus old mouse thing was simply that our mice were kind of dull and not that interested in new things. So they did novel toys and all of those kinds of inanimate object novelties, and found that the mice were normal in their interest in exploring new non-animate things.
Do we have any other questions here? I have one more for you. In that one picture you showed the FGF-8 Neo mouse, and it had the single vesicle. Does that share any other features with holoprosencephaly, like a single tooth or the eyes being displaced?
Yeah, the FGF-8 mutants well, first, the phenotype in the face is variable penetrance. But at its worst, you get midline clefting. So it's a bit different than the sonic hedgehog. You actually get this very horrible midline clefting, similar, I think you get with manta rays in your gene. Yeah, they can get a midline cleft. But yes, but it's a bit different than the hedgehog mutants.
Okay, well, thank you. Let's move outside and see the questions.
The UC Davis M.I.N.D Institute began in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, learning disabilities, and other brain disorders is helping children achieve their fullest potential. Please call or visit our website to find out more about current studies, our research team, and upcoming events.